Bom dia a todos. Hoje a palestra da disciplina Seminários de Zoologia, do curso de pós-graduação em Zoologia do Museu Nacional e do curso de graduação em Ciências Biológicas, ambos da UFRJ, será sobre espécies crípticas e seu significado evolutivo, ministrada por um dos expoentes do tema, professor Torsten do Museu de História Natural da Universidade de Oslo, na Noruega. It's a great honor to have Dr. Struck here with us to talk about cryptic species and their evolutionary meaning. Thank you very much, Torsten, for accepting the invitation. Dr. Struck received his PhD from the University of Osnabrück in Germany in 2003. After this, he has had different positions as researcher and professor at the same university, as well as at the Zoological Research Museum, Alexander Koenig. I'm sorry for my German is not good. Also in Germany and at Auburn University in the United States. Since 2015, Dr. Struck is professor of evolutionary genomics and the Helmet collection at the Natural History Museum of the University of Oslo in Norway, where he's a leader of the research group Frontiers in Evolutionary Zoology. And it has a very nice Twitter account that I have seen that is there in his first slide. And supervises bachelor, master's, and PhD candidates in genomics, phylogenomics, phylogeography, taxonomy, and bioinformatics. Dr. Struck has an outstanding career with influential studies on evolution of Annelida and Spiralia, phylogenetic and phylogenomic methodological issues, and theoretical issues such as the one on cryptic species evolution, most of which are published in high-impact journals such as Nature, Evolution, Trends in Ecology and Evolution, and Systematic Biology. His main research interests are, in his own words, evolution and biodiversity of marine spiralians at all taxonomic levels with emphasis on annelids, using approaches such as genomics, phylogenomics, phylogeographic, and population genetics and bioinformatics. So thank you very much, Dr. Torsten, for being here with us. Without further ado, here is Dr. Torsten Struck.
Yes, thank you very much for inviting me all, and especially you, Joanna, and good to see you again. I don't know how familiar the people are. We have met in Auburn. We were there at the same time. So today I will talk to you about the evolutionary significance of cryptic species, and can put that in quotation mark because I don't think it's the case, a purely taxonomic problem. I don't think so. So when we look, so why is the screen not going on? Let's see now. Yeah, now it's going on. One thing I have to put it, I prepared the talk on a Mac, and when I just went through that before I joined you, some of the pictures will not be very nice, but I think it won't, and some of the font types have been changed, but I think it won't prevent us of our understanding what I want to tell you today. So we have this amazing biodiversity on Earth. Here you can see everything from worms to vertebrates, fish as an example for invertebrate, not a human or a land animal. I'm a marine biologist, so that's what you mostly will see. And humans always wanted to classify this and also name that. And for this reason, I probably think, and we can probably safely say that taxonomy is the oldest biological discipline. Humans always name, for example, animals, plants, fungi, but scientifically it really started, it started before him, but the foundation was really laid by Linné. That's why I put up is Regnum Animale here, because I mean, we are now in a seminar on zoology. And so taxonomy was very old, and taxonomy was actually plays a substantial part in Darwin's Origin of Species, if you ever get the chance to really read it. Many of his arguments are based in taxonomy. But taxonomy has a way, a little bit the view of being old-fashioned. However, in taxonomy, always new problems arrive, or maybe older problems in a new disguise, but at least there's something new. And one of these is cryptic species. Here, for example, cryptic species is not something which are found in some weird unknown species. Here's an example from 2016 by Fennessys and co-authors, where they found cryptic species in giraffes, where they found, okay, what was supposed to be one giraffe species in Africa, it's actually 4. So there's this new taxonomic problem, cryptic species, and that gained a lot of momentum and interest. When we look, this is partly linked to when all the barcoding initiatives started and molecular taxonomy started, because, for example, Fennessy et al., also the recognition of the 4 species is based on molecular data. And this all started with the advent of these molecular taxonomy and the barcoding approaches. And you see here, we did a review paper on this issue and a trend paper. And this is a paper you read in preparation for this one, the tree paper. And there you should have seen this. So here, the number of papers using the term cryptic spe with a star to include speciation increased substantially. And in a large amount. This is a little bit older, but it's still the case. It's still growing. And this has been recognized by a lot of papers already. And many papers dealt with mostly the ecological relevance of cryptic species. As you can see, cryptic species as a window on diversity and conservation, global climate change, species connectivity, or connectivity between assemblies, paradigm shift in species concepts. And there are also a few which also highlighted a little bit the evolutionary aspect, but they are relatively little in comparison to the importance which has been often pointed towards ecological questions. And this you can also see where these papers were published. Molecular ecology, for example, or nature climate change. So what is a cryptic species? I mean, if we speak about cryptic species, there should be a definition, and there's one which is now a little bit more, or a little bit older than 10 years, by Bickford et al. It was also published in Trends in Ecology and Evolution, and it's a relatively straightforward and easy definition. They define cryptic species as 2 or more distinct species that are airlessly classified and hidden under one species name. So that sounds easy and relatively straightforward. However, in this talk, I hope I can show you that this is not the case. And because there was already this debate about cryptic species, are there something real or not? That's what kind of got us going with, this is the product of a journal club I initiated in Oslo when I arrived, and that came into the paper. And for this paper in Tree, we analyzed a little bit more than 660 papers, which we randomly picked from the pool of cryptic species papers and looked for different characteristics. And this is the first part which I will present you before we come to a practical example. So the one by Bickford et al. Was the most often applied definition. So that's the one which is probably the most established at the moment. However, that does not say much, because only 14% of all studies we investigated actually applied the definition at all. Many didn't have any definition. If we did not only have the Bickford et al. Criterion, or if we had other criterions, what did they consider? Other things which often were considered, or at least implicitly assumed, were morphological or phenotypic similarity, or genetic divergence, or that there should be reproductive isolation. Some also include that they should be in the same geographic area. However, there was also a blooming of additional terms: pseudocryptic, semi-cryptic. There's an older term, sibling species, which was coined by Meyer, which has the same meaning, but there were also then that there were hypercryptic clades and megacryptic clades, so this is all there. So there seemed to be a lot of confusion. And I want to give you now a little bit, a few examples to show you this, why there is this confusion. And this is based on examples from my own lab, so that you don't get the feeling we want to ditch anybody. It's just to make the point clear. So this is by work by Ashma El-Ghetani. She was in my lab for a while. And we analyzed Morphometric aegypti. And what we find, and here's one of the examples I mentioned before, where the quality of the figure got really bad to the switching from Mac to PC. They were before considered to be part of Morphometric sanguinea. However, with the genetic data, we found that they were clearly different. And we also looked into the morphology. And when we did that, and Ashma looked into more detail, we found quite a lot of characters which actually are different in Egypti from Sanguinea and also other species. So we could also morphologically, without problem, describe it as a separate species. However, if we would have applied the Bickford et al. Definition, we could have said these are cryptic species. Even so, they are, that was probably just weird taxonomy, which was done there, to be so bold to say that, or before. Another example we actually have here is from Samani Pasocki from the Persian Gulf. She found there Spirobranchus species, and there we have the same story again. So this is Spirobranchus. When we use molecular data, they were clearly separated. This didn't come quite out of the blue, as like for Sanguinea, there was the same. It's not out of the blue. It was expected that these are species complex, and probably there are more than one species beside it. Molecular data again showed they are clearly different from the actual Krause. And again, looking into detail into morphology, we find several morphological differences. Given the Bickford et al. Criterion, could be a cryptic species. Now, in the different example, this is also with an annelid, it's Polygordius, it's an interstitial species, which I did with Patricia Rami Balci together, or which were led by Patricia Rami Balci. And there we find, for example, Polygordius lacteus and Neapolitanus. At the adult morphological state, they are completely identical. She could not find any differences. She's the expert on that group. The same is with Polygordius juani and Tristinus, also as adult, completely identical. Molecularly, they are clearly separated, and they have always been described as separate species. You always found Lacteus and Neapolitanus and Joanni and Tuckerton. Okay, for Lacteus and Neapolitanus, there was at least one criterion. They have different larval types, at least what is assumed. It's not known for all records where they occur, but this is assumed at the moment, and that seems to fit the tree. However, for Joanni or Tristinus, we don't know anything about the larvae. But the most separation for these was always based on geography. It was never really based on larval types. So they are separated by geography. So even so, these species are morphologically identical. We couldn't, if we would apply the Bickford et al. Criterion, we couldn't call these cryptic species because they were never treated under one species name. They were always seen as different species. And we just confirmed that with molecular data. It's not only us who saw that, there's quite a lot of debate about it. And there's, for example, an article in 2017 by Corsuno et al., where they looked at nudibranchs, one group. And what they found is, again, external morphology, they could not separate them. You see they are quite diverse in their external morphology. When they used genetic marker, they clearly split it into 3 different species. When they looked at the radula, they again found morphological differences. So there they could separate them. So what they concluded, there might be different levels of crypticity we should take into account. And that might lead to different taxonomic conclusions, depending on what level of morphological or even molecular level we look at. And then they concluded further in the discussion that we should avoid the term cryptic or pseudo-cryptic species as a reference to a natural phenomenon, so that it is just what they said, it's just a temporary formalization problem of the problems with the delineation of species from the same geographic region when those species demonstrate significant molecular differences. And what they then also said, so for this temporary formalization problem, we should use the term cryptic species. For others, and the term pseudo-cryptic species should be abandoned altogether. So they came to the conclusion, cryptic species not a natural phenomenon. However, there were other papers, for example, by Pauline and Perez Pons de Leon, who looked into parasites and in freshwater. And they, for example, found that in freshwater, we have much more cryptic diversity than in terrestrial habitats. And from them, it seemed to be that there's something linked, that cryptic species indicate something. And these findings suggest that the greater heterogeneity and fragmentation of freshwater habitats may promote higher rates of genetic differentiation among its inhabitants, a general pattern with serious implications for freshwater conservation biology. So again, going back to what it means. detecting cryptic species, and what implication does it have? So the question in a way is it really, are cryptic species a natural phenomenon or not, as Corsuno et al. Suggested, so that it's just a formalization problem. So to answer this, we have to find out which are only taxonomic oddities and which are truly cryptic. However, the problem is the definition of Bickford et al. Doesn't work in this respect. As I showed you before, I hope, with the examples I gave you, we could good morphological species call cryptic species based on the Bickford et al. Definition, whereas others we could not, which might be more closer to what most researchers would consider a cryptic species, that it is morphological so similar that we can't tell it apart easily or at all, without maybe using molecular data or some very sophisticated statistical tools. I mean, there is also then, if we have that and can differentiate that, and if there are cryptic species a real natural phenomenon, which I personally think they are, then there comes a lot of different questions with it. Which cryptic species are the result of recent speciation, parallelism, convergence, or stasis, and how common are these different types? So these are now questions relating to evolutionary biology. Also the next one, what are the relevant intrinsic and extrinsic factors affecting morphological evolution? So what drives the evolution? And to what degree do these factors then affect the phenotype of the cryptic species? If we go to ecology, then are there more cryptic species in certain branches of the tree of life, associated with certain life histories, generalists versus specialists, or parasites, or in certain habitats that we had before, with freshwater versus terrestrial? Is there something in that? And also, how do these cryptic species then affect the composition and stability of ecosystem, and vice versa? How vulnerable are cryptic species to these effects? This comes then towards the direction of conservation biology if we don't take them into account. Does that influence our estimates and our conservation measurements? So what we saw is it's clear we need a clear-cut definition. And one thing we were thinking about in our journal club is that the problem at all definitions as they are is that in a way they intermingle two parts. There's one part which is first we have to delineate the species, and Bickford et al. Intrinsically bring that into the definition if they say it was one before and several afterwards, because there we have that there were differences in distinguishing the species. So that's the first part that needs to be done, but this we need to do for anything, independent if it's cryptic or not. We need to establish if they are distinguishable species or not. And this we should do whatever is best practice for the group at hand, or what species concept we apply for whatever. That's something completely different. Only the next step, after we have done this steps, can then be to ask, is this now a cryptic species? And therefore, we should not link it back to the previous step, where we aligned that to if it was one species, two or three species before, or whatever. We have to do that independently of this. And therefore, we suggest that it should be done because what everybody more or less had in it implicitly was that there is this similarity, this high degree of similarity. So, and put it on a broader perspective, not just for morphology, what you should show is that there's statistical lower degrees of phenotypic disparity than non-cryptic relatives given the time of divergence from the most recent common ancestor. So that they are at comparable timescales. So essentially, if we look at this, so we need to get out of this green zone. We need to be clearly in these areas where we are certain that these are different species. And as I'm aware of all the problems associated with that, but that's a completely different problem than assigning the status cryptic species. That's a problem which has to do with the species delineation. And then we assess the phenotypic disparity, and then it needs to be in this region here, in the blue region. So it needs to be clearly lower phenotypic disparity than we would expect given other non-cryptic species. This has several advantages. It's property-based. We're comparing here disparity indices against divergence or time. It's independent from taxonomic tradition. It doesn't mean if there's a tradition of lumpers or splitters, because we don't care about if the species have been described before as one or two. It just matters how similar they are morphologically or phenotypically to each other. Also, levels of crypticity become obsolete as we can relate them either to divergence or time or to disparity levels, instead of saying, oh, we compare crypticity, pseudocryptic, hypercryptic, megacryptic. And we can use the same categories also then to conduct meta-analyses based on these different levels, and don't have to take cryptic species at face value because just an author said it's a cryptic species. Because we come to this, what we also looked at is how good were these studies done? So how good is the empirical rigor of this study? And that is actually needs to be improved a lot. So when we look at this, only 35.5% of the studies were based on more than one molecular marker. And most often when it was based on only or still 1/3 were based on, 1/3 based on. This was mostly uniparally inherited one. So why does it matter? Here's an example from an earthworm by Gisser et al. From Poland. When they analyzed that from 4 populations, they found different clusters, which they grouped together at species. So they found 3 different species, which they said, okay, we have cryptic species. Also in the network analysis, you can see, and all of these occur in sympatry. So kind of indicating, yeah, that's good, because they occur in sympatry. It's not an allopathic population structure we see here. So they are based on mitochondrial data, and they have high genetic distances between A, C, and E, up to 16%. And then based on this, they said we could conclude there are 3 cryptic species. However, they went further. They also used DDRAT, so birental the nucleus. They probed the whole nuclear genome with a reduced representation method. And when they did this, you can see this here. It's the same specimens as with the mitochondrial marker. And they neatly separate now by population. Also, when they do admixture plots, so where they measure how much gene flow or shared variation is between different species, again, they neatly separates based on population, on site. So individuals group based on site, not on mitochondrial clade. So actually, the nuclear data showed there are no cryptic species at all. So we need these kind of data to be really certain. The other thing is everybody implicitly said, oh, they're so similar, that's why we cannot separately. Only 40.27% of the studies included explicit analysis of the morphological data. And for us, explicit analysis of the morphological data was that they looked under them on the microscope or something. So that they at least looked at the animals and just not take it for granted and maybe made pictures. And only roughly one quarter looked at other phenotypic traits. So also on the phenotypic side, is actually, we need to improve much better. And then there's a lot of debate on that cryptic species are just young species, that not enough has, we looked into this, we cannot, this is percentage based on all studies, but many studies we could not judge if they were young or old, but what we can see roughly, what you can take from it, there's as many young as there are old species. So it's not just a phenomenon of just recent speciation. Then we put out certain criteria, and 15.4% of the studies combined several different types of molecular markers with morphological and or other phenotypic data. So, and compared the genetic divergence of the cryptic taxa to other generic non-cryptic species. So it's very little, which would fulfill the minimum set of the criteria we put out now. If we go even further and say, okay, we also want to know the age to be able to set it in relation to time, then it's only 2.6% of the studies which do that. And if we would say, okay, genome scale data should be used, then we are below 1% of the studies which apply the criteria. So which processes are discussed? There's recent divergence. So that means the species have just evolved and didn't have enough time yet, or it is not fast enough to accumulate morphological differences. You can see here, if we compare the cryptic species versus the non-cryptic one, given in a trajectory, it might actually not be so different from the non-cryptic ones. Other cases, which is a really interesting phenomenon, but where I won't go into today, is that there might be either parallelism or convergence. So that both essentially mean that there was an independent evolution of the same new character suits. So that these evolved, that they're actually in completely different groups. And independently, they are not sisters to each other. These are the blue dots here. And they evolved independently from each other, the same phenotype, which is very interesting because that must be a very strong selection pressure towards the same character traits over the whole species. The phenomenon which I will go into, which I'm more interested in, is stasis. And this is the retention of same ancestral character traits over extended time periods. So you see here, this like D1, T2, they are relatively similar in age to the split from D1 to D6. And while D1 to D6 accumulated quite a lot of differences, D1 to D2 did not change that much or not at all. And I will go to this much more detail now for the rest of the talk. So to sum up this first part, a new definition of cryptic species is needed, in our opinion. We put it out. Species have to be distinguishable, but that's the standard procedure. And if you want to assign it as a cryptic species, then you have to show that it is exhibits statistically lower degrees of phenotypic disparity, or at least morphological disparity, than non-cryptic relatives given time of divergence since the most recent common ancestor. So that they are comparable in time scale, or genetic divergence if you can't work with time. The other thing is, which we also find we need improved rigor. Ideally would be genome scale data. It's getting easier and easier, but I also know that's a challenge for many countries still. But what we can always do is explicit analysis of the phenotype. Nothing is preventing us from that. And also these phenotypic variations should be explicitly quantified to some degree. And then we should test the degrees of phenotypic disparity if they are significantly lower than comparison to non-cryptic species. And ideally, if possible, we should put in a temporal dimension to it. So at least the points 2 to 4, I think nothing prevents any researcher in the world to do that, maybe except for lack of experience with the group. But this everybody can do. Okay, now I want to go into the example, and this is based on Serka's PhD thesis. He finished this year, and he did most of the work, not all, but really 80% to 90% of the work. And we will talk now about the Stygocapitella species complex. So what is Stygocapitella? It's an annelid. It's rather small, less than 3 millimeters in length. It's very characteristic. When you want to find it, it has here a whip-like kita, which you can recognize it. But when you see it first with a microscope, you might maybe think it's an Amphitrate, so an oligochaete. It's gonochoristic and has eternal fertilization. It has direct development in gel package, which attaches to the sand grain. Why I'm telling you this? Because this has implication for its life cycle. There's no pelagic stage in this animal. So it doesn't distribute via the pelagus. All in all, it seems to have, at least from what we know, very limited active dispersal capacity. Where do we find them? They occur in interstitially, so between the space in the sand grains on sandy beaches. And they're around the high water line or higher, and then at the depth of 10 to 90 centimeters. So even from there, getting passively transported into the open water column is not very likely. They're very high up the beach, or relatively deep. Nonetheless, we find a global distribution, except for the tropics and subtropics. And here's where they have been recorded as one species, except for the dot in South Africa. So that's where all was one species, Stygocapitella subterranea. And with that, there were a classical example of the Meyer-Fauna paradox. I won't go into this. If you want to read more up on that, look in the paper by Jose, his review paper on that. We did not go out in the blue in this. There had been in 2004 a study by Smith and Westheider where they took one population from both US coast and one from the US European coastline, one population per coastline, and found with rapid data that the populations are reciprocally monophyletic, maybe possibly indicating that there's more than one species present. However, there could also be just population structure. I mean, these are very far separated from each other, low dispersal capacity, so maybe that's just it. Now again, what's not so nice to see, I was in South Africa sampling, and there I found a new Styrocapistellar species, and when I looked at in the morphology, there was ample morphological differences, and then we started to study it more. You can't see that so well, but where's the mouse there? This is a minuta. Where's it? Styrocapital minuta. It's here. It lacks all these small chaeta, which are indicated here. It's called 4 chaeta. It doesn't have them at all. It has only 8 chaetigers instead of 10, and it's substantially smaller, or at least we thought it was substantially smaller with the sample we had at that time. They were also clearly morphologically, molecularly separated, and this is not CO1, this is 18S, so we could separate them based on 18S. And the other thing is morphological disparity nicely correlated with 18S disparity. So that seemed all pretty nice. And well, we described them as 3 different species because we also got in samples from Australia. There. And that was kind of the starting point for José's work. There we extended it now substantially. We involved more than 350 specimens from 36 populations all over the world. With a new record from the Russian Pacific coast, we use still their classical approach with 4 molecular markers. 2 are mitochondrial, 2 nuclear, so at least to fulfill the criterion that we also collect from the nuclear. When we did this is a combination of all combined, but single gene tree analysis shows the same. We find in the end that there are 12 new species, or that we can split them in 12 species. Sorry. And 9 of these were new to science because 3 we already had described before. 8 of these species we could formally describe, 1 we could not. Sorry. Because we lacked a phenotype, specimen to deposit as a holotype. 7 of these 9 species were formally assigned to Styrocapistellar subterranea, but 2 were not. So there we have this conundrum that we could say, based on Bigfoot, some were cryptic species, while others were not, even so they were nested within these cryptic species and looked morphologically the same. When we look at the morphology, we find only 4 morphotypes. There's the one which is not shown here, which was before, the South African one, which is quite different. But then we also find these 3. This is the one, the canicle, the Styrocapitellar pattern, and here there are some slight differences in the chaetal patterns. I won't go into detail. It's very minimal. It's just the differences between 1 or 2 chaeta which are there or not there. And that's all which we found based on these discrete characters. We also looked in morphometric analyses. When we analyzed them all together, you could not statistically separate them. However, when we went by morphotype, then these we could not separate from each other statistically with body length, and the others show the same pattern but have less statistical power. With this blue morphotype, we could separate 2, but on the other hand, there's also overlap. This is bridging them. So it is kind of a continuum. And here we could separate 1 from 2, but these 2 are inseparable. We also looked then with principal component analysis, and you see when we take them all, they clearly overlap, except for the South African one. When we go in the morphotypes, you could separate them. But you also see as soon as we included more individuals here, very few, they start to overlap again. So it's really hard also there to differentiate them. You can tell them apart, but in a way that's afterwards going with our molecular knowledge on it. So by itself, it's really hard to separate. So considering morphotypes and morphometrics, some species can be differentiated, but the species are still, even if we do that, they're still extremely similar. So much similar than we would anticipate. So here's a summary. We have 12 species now. Here we see the differences in the chaetal patterns and also some in the number of chaetigers, what we can summarize up. This has effects for the distribution. This is from a review paper we are just preparing for the whole family, Pyrgotelidae. and Orbinidae included. We see here, you see all the black dots is now all the records where we don't know actually what species that is, because we need our molecular data to be able to assign them. And they were also only described without detailed morphological description. Then here we see there's several colors at the US East Coast, and there are several colors here at the Russian, at the US Pacific coast, also at the Russian coast here. Also at the US East Coast, we see here there's two colors. In Europe, we see several different colors here. If we go into a little bit more detail, we see they're either very closely related to each other here in Europe, or even here in Musselburgh. All three species occur at the same beach. They occur in sympatry, and here I mean really sympatry. We found species within the same 50 cubic centimeter of sediment. And it's in Sylt, we find the same. Here it's on Friday Harbor. We find on Friday Harbor itself 4 different species. It's a tiny island in comparison on a global scale. And also here on the East Coast, there's one species which usually only occurs in Europe. We find 2 representatives also here in sympatry in Lübeck. Also, the habitat was expanded. They occur in habitats which wouldn't have thought they would occur. The first 2 rows show the typical habitat, like nice beach, not quite as nice as Copacabana, but you're going in this direction. But we also see now that we can find them in these, in these beaches here, where we have pebbles on top, but which are still relatively large, but also in short beaches like here, and which do quite different. So they occur in the habitat range. It also seems to have a range, but there's no correlation between habitat and species. As I said, they're kind of. Then we went a little bit further and looked now more towards the evolutionary stasis point, and really looked at the temporal dimension of this. The first thing is we compared the degree of Stygocapitella towards other families. Orbinidae is a sister group to Pygotrillidae, and Nerilidae is another interstitial species, which is also relatively closely related. And you see here they occur much larger morphospace than Stygocapitella does. When we again calculate the multidimensional morphological disparity index, the pairwise one, we see that Stygocapitella is much more similar to each other than these 2. One could say, okay, one is the genus, the other is family. It's not surprising. And here we also these were data set generated for genera, so we were comparing genera. Here we are comparing species. Despite the fact that these hierarchical levels are artificial and man-made, we compared that against genetic distance to get a temporal dimension on that, but we also did molecular dating. And then you see now, while here, pairwise genetic distances, the morphological disparity increases relatively fast at very little pairwise genetic distances. In Stygocapitella, it remains unchanged. So actually, we can compare these to each other. These are reasonable pairwise comparisons because they span the same genetic distance in their comparison. And this is this is based again on 18S, not CO1. And we see here it's flattening out, indicating that more or less the whole morphospace has been occupied by the samples. And even here, so that's about 1 to 8 times more dispersed than Stygocapitella is. And even when we get up to the higher level, only very few are as little disparate as Stygocapitella is of the other comparisons at that level of genetic diversity. We also mapped then the morphotypes on a dated tree. And here's just a summary of that. So we don't know when the first or the red morphotype evolved, so the one which is found in South Africa, but we know for certain using different reconstruction methods, they all give the same result. So here we have morphotype 2, the green one, and that have to have occurred at least 140 million years ago. Then we have morphotype 4, which occurred or evolved at that point here, it's 65 million years ago. And then even that one, the blue one, which is the youngest, but young means here 80 million years ago. So for morphotype 2 and morphotype 4, the dinosaurs were still around. So, and since then, they did not change their morphology at all. So this is often referred to as living fossils, as we don't have fossils in our group. They can't be living fossils. The other thing is living fossils often, like the example here, that's a coelurant, refer to character traits and not the whole specimens. Crocodiles is another example of living fossils, and fossil crocodiles have a different genus, often different family names. It's referring to character traits. However, we also have that for species, and that's in the paleontological realm, and it's a hypothesis from Gold, the punctuated equilibrium, where we also have time series which don't change, which are the same species without speciation. And there we have this stasis component. And then, as I said before, we have this seemingly no evolution in cryptic species. And what they all link, we find that in here, and what's also in here is stasis. That's a common ground. And cryptic species are, I think, a good way to look into this phenomenon, because they, have they affect the whole species, like here, the paleontological record, but are recent. So like this living fossils. So it's something where we really can work with genome data, for example, on. So what we have to think, and this is a slide from Jose, which I very much like, and that's why I included it. What we have maybe to start to think about is now about evolvability. And on one extreme, we have stars, on the others, we have adaptive radiation, and then everything in between. And there's a lot of research going on the right side of this slide. There's a lot on Darwin finches, on Glycid. They are the big issue in evolutionary biology. That's what's studied. This one, okay, that's in paleontology, but for recent species or extant species, there's really done nothing. On this other phenomenon that nothing happens, or seemingly nothing happens, there's no research, or really no research done. And I think if we want to understand evolvability, we have to look at both sides. Then we might also be better able to understand what's happening in between. So, and that's why we think it's a good idea. And we started to probe into this a little bit, and I'm presenting now data which are under review. And these are the courses where here's a review paper by Jose and me, where we discussed courses for stasis and how they relate to cryptic species and how can they be tested. And I just briefly go to this. So it's stabilizing selection, where we have different factors which can cause it. For example, that the niche is very conservative and doesn't change much, or that the niche changes its positions, but the species can follow that positional change. That can be the optimum might fluctuate or around a long-term stable mean, and other sources. The others is constraints, so that's something intrinsic. Stabilizing selection is extrinsic, it comes from outside. Constraints come from inside. So that's, for example, pleiotrophic effects, so that the gene networks just do not have an effect on one, but also on another part of the body. And for that can't change, because change in one might have a negative effect on the other part. There might be low standing genetic variation, limited rate of evolution. The interesting is there might also be a high degree of variability. There's so much variability on the morphological level that we cannot differentiate interspecific variability from intraspecific variability. And that will also cause for us cryptic speciation or cryptic species. The last component is that there might be shared genetic variation due to admixture, so that there's hybridization still ongoing. Even so, there are different species. We see that more often occurring, even in animals, that there are hybrids, even between distantly related species and old species, or that they carry on incomplete lineage sorting, and that somehow contributes to this. The thing is, you should have in mind, all these categories are not mutually exclusive. They may actually influence each other or necessary for one and the other and can act at the same time. So that makes it a little bit harder to maybe tease them apart, which you should start to look at this. And we started now to work on this, giving it a genomic perspective. And now many of the very bad pictures come, but if we just go by color, we should be able to see that. I mean, that's what matters here. On the left-hand side in the tree, you see that's still our 4 nuclear markers, the 4 molecular markers. And on the right-hand side, it's DD-RAD data, so probing the whole genome. We see a general congruence. You see the colors green, blue, and red are generally congruent. But if you look carefully, you see 3 species are placed differently. They're indicated here with the arrows. So there's a slight incongruence. However, the populations which are affected there are not sympatric populations. These are populations which are allopatric, meaning there at these populations so far we know only that there occurs one species and not several. We also looked at a network, but sometimes that might be better representation, especially if we think about hybridization. What we see there now is that only one species remains placed differently. The other ones, these are the ones indicated here with the arrows again, they are not grouped with the others, but drawn towards the center. And they are not only the ones, there are also other species here, which are a little bit more drawn to the center of the network. And this is especially the case for Styrocapitella Maria branchi. There are a few species which are further drawn towards the center. We also did different plots. This is a PCA, and this is a multidimensional scaling plot. Or you see it, not me so much. Again, the colors are relatively similar. Here are the three different species. One is again placed differently, and then from this, from Maria branchi, some are drawn towards the other ones. We see here in the MDS, the lower one, which is more important, there Styrocapitella Maria branchi is more spread out, whereas the other two are more concentrated, and one again is placed differently. That one sticks out. The reason for the differences in these two plots is that PCA is more susceptible to missingness, and that's a problem we had in our analysis, and that pulls specimens to the center of the PCA plot. And so this is probably more the realistic representation of it. So in a way, this fits what we see with the network, that there are from Maria branchi, that there are some which pull towards the middle. We also looked at admixture, and what we can see here is now, in contrast, if you remember the admixture plot from the earthworm, here we have shared genetic signal across all three species. When specimens, when we see several colors in a specimen, that means that's shared. There's a shared signal between the different species. And especially, and the least we see in the species Styrocapitella westidi, which is on the US coast. Sympatric populations are less or equally affected than the allopathic one. So if it would be recent gene flow, we would expect that the sympatric populations are much more affected than the allopathic ones, but that's not the case. Again, this one individual, we don't know for what reason, but it's always this one individual, which is good that it's so consistent. It sticks out again. That one grouped groups with Subterranea instead of Maria branchi, where it is with the 4 genes. We also did modeling of gene flow, and here are just values. And the first thing is all these models are these are models of recent gene flow, and none of them is the best supported. These are the 3 best supported scenarios in all, and these are all the models which include recent gene flow. None of them is the best supported one. And this is also in agreement with other measurements we did, F3 statistic, and that there's no increased polymorphism in sympatric populations. The 3 equally well supported scenarios generally follow the phylogeny. So there's either one is there's no gene flow at all, then there's gene flow at this node, and then there's gene flow at this node, which is kind of indicating that there's ancient gene flow maybe going on with it. The truth is we can actually not differentiate ancient gene flow from incomplete lineage sorting. So it could also be incomplete lineage sorting at that stage. What we also looked at, we have no indication of bottlenecks at the population level. So it's not one hypothesis that reoccurring bottlenecks cause that the phenotypes always stays with the old phenotype, with the majority phenotype. We don't have any indication of that. So what's left for us now at the moment, at least from the genetic level, is that it might be a possible reason could be ancient gene flow or incomplete lineage sorting, which is still dragged on to recent specimens. And that kind of maintains this morphotype. All these 3 Atlantic species have, by the way, the same phenotypes, the classical Styrocapitella subterranea phenotype. So they are all the same morphology. We're also working on the genome, and I think I just skipped this now due to time. We are working on it. It's a hard case because it's much bigger than we anticipated. It's 2/3 of the human genome. we will have to work with a small one, so we don't get much out of it. And especially the PacBio stuff causes some problems there. But there have been some great news this week, which might help that we will be able to solve that in the near future, given some developments with PacBio chemistry. Okay, to come to the conclusion for this, Stygocapitella species complex consists so far of 12 species, not just one. Some species exhibit slight morphological differences, but even these are extremely similar to each other. Our species are morphologically, other species are morphologically identical. We can't differentiate them at all. Species did not change their morphotypes at all for tens of millions of years, up to 440 million years, and maybe even longer. And Stygocapitella species show an extreme case of stasis and an ideal model to study stasis using neochronological species, so extant species. Recent hybridization and bottlenecks can be excluded, ancient gene flow or incomplete lineage sorting a possibility. Shared ancient signal might be the case. Now I want to wrap up with a more broader view, what I think why it is good to work on cryptic species. They have a high potential for multidisciplinary research. And this Jose and I wrapped up in a review paper in ELS. So here we have taxonomy in a way, so the delimitation process. And with that, it helps us understand evolutionary processes like stasis, recent divergence, convergence, parallelism. So that can help us a lot, improved understanding of these processes. On the other hand, we also have an improved understanding of genomic and transcriptomic data by this, due effects of civilizing selection due to this, but also on the other hand, eternal factors back on stasis. And cryptic species can help this. Genomic data can help us to improve the databases for the taxonomy part. And both of these levels can also help on the next level. So here we are kind of at the species level, here we're at the cellular level, and then we go to the ecosystem level. Cryptic species can improve diversity assessment. On the other hand, this can also help us again with the delimitation process. This can help us with understanding external factors better. On the other hand, the changes in time and space can have an influence on how we understand macroecological processes. If we know that maybe some species react not very fast to changes and are relatively constant. And the one thing you have to think about, for example, with Stygocapitella, but also with other cryptic species, if they have been around for 140 million years, they have experienced quite some environmental changes during their history now. And that brings me to maybe some more provocative questions. What is evolutionary success? Nowadays, everybody speaks about huge evolvability and adaptive radiations, that what species need to be. Maybe these long-lasting species, which have sustained changes for tens of millions of years, are much more evolutionary successful. They have sustained the test of time, whereas the others have still to show if they're capable of that or if they don't go extinct within the next 2 million years, including us. Which species may not get extinct in the anthropogenic climate change is, in my opinion, then linked to it. In the moment, it's a lot that the species which don't show high evolutionary evolvability might be this, but maybe it's actually that these ones, because they have sustained so long, have some other properties which help them to stay there. And with that, I want to thank you for your attention, and as well as several people included. So I already mentioned Jose. In the lab, there were also a lot of work done by bachelor students, so undergraduate students. That's actually how the whole Stygocapitella project started. It was bits and pieces with bachelor theses, which compiled the data for me. And then that was so much that I could convince here in Oslo, the museum, to give us money for Jose's project. And then there was a lot of collaborators included through all these stages regarding sampling trips, discussion, the tree paper, that's all the authors from the tree paper. And the good thing is of this came out of a journal club. That was a product of a journal club where we for 1 year spoke about cryptic species. And there were very different people included. We had someone who worked with fungi. We had, or some which work with fungi, with plants. We had people from the paleontology in there. We had genomicists in there. We had ecologists included. We had people working with insects, and then others like me working with marine inverts. So the only people we were lacking were vertebrate people. All these people brought very different views to it. For example, as the taxonomists often spoke about morphology and that it doesn't change, but then often came, yeah, but there's more than morphology to an animal or a plant or fungi. It's a phenotype and things like these. And time with paleontology was very important. And I think for that, it's good to have different people look at a problem. And journal clubs can be very productive. And with that, I want to stop this part of the presentation and stop sharing.
Thank you very much, Torsten, for the great talk. And that's also to all. That's all to all that are watching the talk with us on YouTube, and I hope you enjoyed as much as we did. Muito obrigada a todos que estavam por aqui assistindo com a gente no YouTube. Espero que tenham gostado tanto quanto nós. Só just find myself.